Okay, hi everyone. This is the last PowerPoint, and it's going to be broken into two parts. This first part is going to probably take us up to about slide 18 on biofilms, and then the next part will uh, be at the end of this week or even next week. That will be on the final. You will not be responsible for part 2 of this presentation for this exam 4. Okay? Anyhow, this is about humans and microbes again, but we're going to be talking about a different type of pathogenicity, the stealth pathogens. So let's begin with this first slide here. The traditional thinking is what we've got here listed as the 4 bullets, and this is the result of the golden age of microbiology. One of the things was that all microbes are germs, and the cleaner we are, the healthier we'll be. And some of this was introduced in the golden age of microbiology when they discovered, I think it was Lister, was one of the individuals, discovered that using antiseptics cut down infection after surgery. So we've only built on that from there. Another thing was the germ theory, that one microbe causes one disease. So, as a result, that one microbe in humans, if that is eradicated, then in general, the humans or human will be healthier. We have kind of found other things further along that line, in that not everybody is perfectly healthy if you get rid of the one microbe. It all depends upon the host health, as we know, and factors such as our microbiome. The third bullet there is that the human response to infection is predictable, and that healthy people will always develop a long-term immunity. Again, we know that that's not true because of there are some people it does take a long time to recover. The host immune response does play into that. And finally, why get sick with an infection since they've discovered the vaccines, let's get vaccinated. And that will give you the same long-term immunity as the infection. Again, that's not always true. I know that for me, we were told that chickenpox, if we got the disease chickenpox as kids, that actually there was some thought that we'd have a longer term immunity versus those that have had the vaccine for it. Again, I'm not right up on that data, but it isn't always true that the disease will cause longer immunity. So anyway, I'm going to talk a little bit about mortality rates here, and you remember that from epidemiology, that the mortality rate would be the percentage of people that die for a specific disease. Well, in this case, we're kind of making a general definition of disease here, but it's the top 10 causes of deaths in among a population in low-income countries. Now, this was taken in 2016. If you look at that graph, all of those red bars represent communicable or else infectious diseases, many of those red bars. You'll see that there are just 2 bars there, and that is a non-communicable disease, such as heart disease or stroke. And finally, the green little bar at the bottom is due to injuries. So in these low-income countries or third world countries, the majority of deaths are caused by infectious diseases. And these are the numbers. So the mortality rate from all diseases or deaths in a country such as this low-income country is 440 per 100,000 people. Okay? And the total deaths, though, are 252 per 100,000 people, will die of infectious diseases. Now, is that a case of sanitation, hygiene, or poor economics that treatments are not available? It may be one or all of those. But if you compare this table to the next slide here, in high-income countries such as the United States, you'll see that most of these deaths are in blue. Most of these deaths are due to non-communicable diseases, such as heart disease, stroke, Alzheimer's, cancers. You have only, down there about midway, we see that lower respiratory infections are a cause of death in the higher income countries. If you look at this, you've got about 470 deaths per 100,000 people. That's the mortality rate of deaths per 100,000 people. Interesting enough, that's about the same mortality rate for the lower income countries. It's about 420 per 100,000 people. But in this case, the deaths are due to other things instead of infectious disease. Okay, we're much, much lower as far as deaths per caused by infectious diseases. So again, is that due to the economics, the distribution of treatment, sanitation or hygiene? It may be about among all these factors that contribute such a difference in the locations as far as the causes of death. What we do know, though, is infectious diseases, some of them do cause death. They do have a profound impact if you do acquire an infectious disease on the human microbiota. We know that now. And not only is the disease impacting the microbiota, the use of antibiotics, other immunosuppressive drugs, and new technologies affect the rate of recovery from these infectious diseases. And again, it does that by affecting the human microbiota. Okay, and once we've got an imbalance with the human microbiota, they are now finding that there's more of a link between that microbiota being in dysbiosis and the complete cure or healing from an infectious disease. So now this is what we have as a model of infectious diseases in the microbiome age, because it's all about the microbiome right now. But you'll get an infection which may lead to inflammation, and then, or it may lead to having an effect on your immune system because that microbiome has been disrupted. And in either case, the inflammation or even the infection itself may suppress your immune system, or it may even dysregulate it. It may kind of hinder it in functioning correctly. Whereas in the past, they pretty much felt with the with infections back in the golden age, that infections cause disease, and then the disease, you'd either, be cured or die. So anyhow, this is just a slide. If you look at this on the right-hand side, basically, under B, we've got a healthy control, and each one of those colors, represents a different phyla, species of bacteria from the gut, and then compare it to the patient, the person that has a disease. And again, the colors are different in almost each of those cases, and dramatically so in the top one and the bottom one. So again, this is disease that has been proven can affect our microbiome, and we know that the microbiome provides a lot of the metabolic processes that we need in order to live. And I'm going to say here that I've talked a lot about the microbiome of the gut, but we have microbiomes, different locations on our body. In the hair follicles, our mouth, vagina, the skin. So any one of these can be affected, or the landscaping of the phyla will change with disease. And again, this kind of, I think, comes from the last PowerPoint presentation. They have been able to measure, I think through DNA testing, the types of species of bacteria that are in individuals that have fibromyalgia and those that are healthy, the controls. And the one that has a fibromyalgia is the diagram there in the purple block. So if you look at that, it's got about 3 times the amount of bacteria, but surprisingly, a variety of bacteria. And the control will have, in this case, I think it's only about 5 different species of bacteria, but these 5 species are not pathogenic. They actually can be beneficial or commensals. So that's just one disease where they've compared the microbiome genetically or DNA wise as the makeup. So anyhow, this is an article that is included in this week's lecture. So give it read it over, please. And this is just talking about patients with Lyme disease. Those that have had acute Lyme disease, you're bit by a tick, you're showing signs and symptoms, you're given doxy. And if you get that antibiotic intervention, about 80 or 90% of us will overcome the infection, will be quote unquote cured. But there's probably about 10 or 20% of people that, although they are given treatment, they will still be feeling fatigue or other symptoms of Lyme disease. And this is known as post-treatment Lyme disease syndrome, PTLDS. And then they really don't understand why that happens. We aren't there in the research as to why some of these people, it becomes a chronic disease. But they have found that these individuals have a distinct microbiome signature. So they have distinct makeup of their microbiome. And in fact, they can even do sequencing, genetic testing on this microbiome, and 80% of the time it is correct in diagnosing Lyme disease. So anyhow, give this, please look this over. They're naming here too in this green box some of the phyla or some of the species of bacteria that are present or increased in number, and then some of the others, such as Bacteroides, is decreased. So anyhow, this is going to be a new way of testing for disease. Maybe making, do gene sequencing as to the microbiomes, and that will be able to determine what disease we have. And again, this is kind of a review from the the fungi on us or in us. And even that, we do have some fungi that normally do occur in our microbiomes, but even that in the state of disease will give us a different picture. We'll have different species of fungi present. And if you look in the right hand side there, I found it rather interesting here. Even in the top box there, it's showing dysbiotic microbiota, and it's talking about HIV in the mouth. The HIV mediated immunodeficiency, so that virus causes severe dysbiosis, which is correlated to the decreased numbers of immune cells, the CD4 T cells. So even with our mycobacteria, even that in dysbiosis can affect us at the immune response level. Now I'm going to begin talking about the pathogens and a little more about stealth pathogens in this PowerPoint. I will continue next PowerPoint presentation talking about specific pathogens, especially the stealth pathogens. So the ones that we've heard of all along are the primary pathogens, okay? These are also known as frontal pathogens. So when you hear primary or frontal, they are the same pathogen. And these are the guys that cause the acute infections. So they have fast generation times. Their motto is get in, reproduce quickly, get out, leave the host. And this is they also carry the genes for toxins and other virulence factors. Okay? And at the population level, after they've reproduced enough, they will express those genes that produce the symptoms, such as a fever or sore throat, muscle aches. So these are the signs and symptoms. This is an example of a primary pathogen. And they usually do trigger a very predictable immune response, such as we get the antibodies produced, the immune cells are activated, and the microbe is cleared from the host, either by the immune system and or it will leave the infection site. And that's usually by, like I had explained a little bit last week, by the way it enters or the way it will release or be released from the host cell. Either through sneezing or other such reactions. And that's what that says in that last bullet. Okay, they rely on the host response to allow them to spread to new hosts. So this is a primary pathogen, typical model of an infection or an infectious disease. Now it gets a little complicated. There's a little more to it, this story. So we do have pathogens that are called opportunistic. Okay, opportunistic. Now these are microbes, are usually present in the environment, or in example for us, they are present on our other parts of our body, and they normally do not cause any problems or any acute illnesses. But if given the opportunity, if they travel to another part of the body, or if the immune system or the microbiome is out of balance, out of kilter, these opportunistic pathogens will now become pathogenic. And here's an example of the Staphylococcus aureus. It causes sepsis when it gets into the blood from the skin. So it's somewhere where it normally doesn't occur. Usually, I think I'd mentioned this, about 30% of us have Staph aureus in our nasal passages. They're there, they are not causing a problem. But once that microbe enters the blood system, it can now become a serious pathogen. Haemophilus influenzae and Streptococcus pneumoniae, they cause pneumonia. They may be present in our bodies, but once they hit the respiratory system and other factors such as our immune, the level of our immune response, they may cause problems. Now, pathobionts, that word there, these are a microbiota that do cause disease at a population level, but they are usually associated with chronic inflammatory conditions. So if we look at these in that table there on the right, pathobionts of the gastrointestinal tract, let's go down to that Helicobacter. It's the third bacteria listed there, Helicobacter pylori. Okay, many people can have these, may carry this Helicobacter pylori, but not all of them will, not all the people that carry these will develop gastric cancers, but they have been associated with gastric cancers. It's just an example. Now we're going to get into the 50 shades of pathogenicity. These are your bacterial stealth pathogens. So I talked about the frontal pathogens. Talked a little bit about the opportunistic pathogens. Think of the word when they get the opportunity, they can cause infectious diseases. Now the bacterial stealth pathogens are another type of pathogens. And what they end up doing is they will affect the immune response in order to promote their long-term survival. Now, they manipulate the human immune response. So it isn't like a knockout fight with the immune system. It's kind of a sneaky way either to avoid or to kind of manipulate our response so as not to cause a outright response to the infection. So anyhow, here are some examples of stealth pathogens. We've got syphilis, we've got chlamydia. Look at the third bullet down there is rickettsia and anaplasmaceae. Those are bacteria, very, very small bacteria that live within the host cell, avoiding the immune response. You've got Mycobacterium tuberculosis, and finally the bacterium that causes Lyme disease. These are some examples of stealth pathogens. These are the comparisons between frontal primary pathogens and stealth pathogens. So please know these. They will be on the exam. Know the characteristics of each. If we look at the frontal pathogens, their survival as a species depends on the transmission to new hosts. With the stealth pathogens, it doesn't matter on whether they are reproduced quickly and transmitted to a new host. They don't need to be transmitted to a new host. On frontal pathogens, they reproduce rapidly. And so antibiotics are really effective in treating frontal pathogens. With your stealth pathogens, they reproduce very slowly, and antibiotics are not usually effective. Frontal pathogens, the body responds by having both the innate and adaptive immune responses kick in. And as a result, we get overt symptoms and signs which help in spreading that pathogen to a new host. Again, like I said, you get congested, you sneeze. That's basically getting rid of that pathogen in order for it to be transmitted to a new host. In stealth pathogens, again, if we look across the way there, it engages some components of the innate system, but usually, rarely, not the adaptive system. So you do not have antibodies forming against these stealth pathogens. It's usually, the most reaction you'll get is usually due to inflammation. Frontal pathogens, the immune system will eliminate it. Infection is usually not cleared by the immune system in stealth pathogens. And finally, there is a long-term protective immunity that is achieved usually. With your stealth pathogens, usually not a long-term protective immunity. So if you just can kind of look over those and know these characteristics, the contrast and comparison between the two, you'll be set for the exam. I'm going to probably ask. I'll have several characteristics and have you tell me whether they are frontal or stealth pathogens. So these are the strategies of the bacterial stealth pathogens. Basically, it's established permanent infection. They like the host. Okay, they're happy just to stay in the cells, many of them of the host, and live there. They will have periods of growth, and again, they affect or manipulate the host immune responses. Some of the ways in which these stealth pathogens work is, first, they grow in biofilms. Many times you'll see these guys growing in biofilms. They can also stop growing actively. They become kind of dormant persister cells. And that is usually triggered by a stringent response, which I'll talk about in a few minutes. They also live. They're smart. They live where they're not going to be right out there for the host immune system. They usually are protected or buried deep within tissue sites of the body. And again, they invade and reproduce inside host cells. Okay? And that even includes the immune cells. So these guys are kind of sneaky. I wonder if any of you remember the stealth jets. I don't know if they still make them, but these were jets that were very fast, and they kind of flew below the radar system, okay? And used in battle. I don't know if they were used in battle, but they could not be picked up by radar. This is pretty much like how these stealth pathogens work. They cannot be picked up many times by the radar of our immune system. So these are, I'm going to be ending here in a few minutes, but just going to talk a little bit about the biofilm again. There will be some questions on the exam about the biofilm again. So, as I've said, there are cells that free float. These are known as planktonic bacterial cells. And when they go to start a biofilm, some of these planktonic cells will adhere to a surface. Now, it can be an inanimate surface, or it can be a animate surface, such as a tissue, if it's within us. And once they attach to this surface, these free-floating planktonic cells, they will adhere and then kind of aggregate or cluster closer together. And that's by that type 4 pili. Then they become permanently fixed on the surface. They will begin to divide and grow and mature. And once that biofilm matures, it has many bacteria. They may be also different types of bacteria. There are usually some type of a tunnel system, actually, within that matrix, within that biofilm, mature biofilm formation. And those tunnels allow the flow of water or fluids that brings nutrients and takes away waste. So again, the biofilms have to be attached to an inanimate or an animate surface, but they also must be in the presence of some type of liquid, usually a water environment. And this bottom picture here is just showing you some of the benefits. What does a biofilm benefit, or how does it benefit? First of all, it's attached to a surface. It cannot be washed away easily. Just the form of the matrix itself, of the body of the biofilm itself, aids in capturing minerals and nutrients. It also is able to retain the enzymes, and those enzymes help in degrading the nutrients. And then it's a continuous regeneration. So you're continuously, cells are continually multiplying by binary fission. And you also will get horizontal gene transfer there. You will get even new types of bacteria. So the biofilm really is like a castle, a fortress. It is very tolerant and resistant to the environment. And this does include being resistant even to antibiotics. Okay, so these biofilms have different bacteria, usually have, may have different species of bacteria, some anaerobic, some anaerobic facultative anaerobes. You may even get some gram negatives, maybe some gram positives, but what you have here are cells that are able to become dormant if the environment does become threatening. And this is what gives them the ability for longevity, one of the reasons. So what happens is, let's say there's a lack of nutrients, or there are antibiotics that are introduced into this biofilm. What happens is the bacteria respond to this stressor. What they do is they produce a signaling molecule, alarmin. So if you look at that word alarmin, it means alarm, and it activates this PPGPP, which puts bacterial cells into a metabolically dormant state. They become dormant. So they aren't metabolizing, they aren't at their regular rate, they aren't producing the proteins, non-essential proteins, they aren't growing and dividing. They are basically just in a coma state. Now, this coma state means that they are antibiotic tolerant. So even though antibiotics may have been introduced, they will not be taken up by the bacterial cell. So in this example, they are not so much antibiotic resistant, they are antibiotic tolerant. The antibiotics will not have any effect upon them because they are not being metabolized by those bacterial cells, those bacterial cells that have been activated by some kind of a stressor. So what happens when that stressor is no longer there is that these bacterial cells that were dormant, the persister cells, will then become vegetative again. They will become metabolically active. So this is one of the reasons why which biofilms are so resistant to treatment. There are other things too that come into play. Is that we've got the matrix is usually a thick covering over the biofilm body, and that kind of the layers of that matrix kind of forms like a kind of a covering in which many things cannot penetrate. You also will have some bacteria within that biofilm that may have become antibiotic resistant. Okay, so anyhow, this is some more information on the biofilm, and this is a very good 6-minute montage talking more about biofilms and diseases, and in which cases you will run across these biofilms within humans. And I really please watch this. If you cannot get into it from this recording, then go back to the PowerPoint slides in Blackboard and access it through that. It is very interesting that this is a new type of thing that we've got to deal with today in the 21st century. So anyhow, that's it for now. I have probably about 13 or 15 more slides to go. I will we'll be adding those later in the week. And again, you won't be responsible for that for exam 4. Exam 4 will be this Friday, but there will be some of this information in the next presentation on your final. Okay? All right, I hope you all have a good evening, and I will be seeing you online over the next couple of weeks. I will still be having the office hours if you're interested. Okay? All right, take it easy and have a good day. Bye.